Good morning, good day, and good evening, everybody. Welcome to the Phenomics webinars. My name is Philip van Gielhausen. I will be your host today, and we will hear Dr. Nadia Al-Tamimi from University College in Dublin, Ireland. And she will be speaking about the identification of QTLs associated with growth and transpiration under controlled conditions at the TPA in Adelaide, Australia. First of all, I want to give you a little bit of background about our webinar series, which was designed as an exchange platform, especially during the recent times where Corona cuts most of the researchers and interested people out from attending to conferences and meeting face-to-face. So we hope that we can compensate for that with our webinar series, at least to a small extent, and give researchers and plant phenotyping practitioners the opportunity to showcase what they have been doing, what they have achieved, and what they are working on. In this regard, of course, everybody of you is welcome to submit a proposal to give a talk inside our Phenomics webinars. Just go on our homepage, which is indicated on the link below on this slide, and fill the form, and then we'll hopefully see you in a few weeks here, also streaming live one of our your presentation. The webinar series takes place every second week on a Friday at 1400 Central European time. The initiators of the Phenomics webinars are the International Plant Phenotyping Network, so that's the place where I work, the European Plant Phenotyping Network project EPPN 2020, and Emphasis, also a pan-European project, but I will tell you a little bit more in the next few slides. This webinar series is being brought to you in partnership with the University of Nebraska-Lincoln and the Agri-X Forum in China. First of all, a little bit to IPPN. IPPN's goal is to increase the visibility and impact of plant phenotyping worldwide, and to enable cooperation by fostering communication between stakeholders, may it be in academia, industry, governmental, or the general public. We are doing this by organizing workshops, for example, symposia, and yeah, we have different working groups, and all these informations are then being bundled and brought to you in form of our website or through our many meetings and events that we are organizing. Of which I'd like to mention the next International Plant Phenotyping Symposium, which is not happening anywhere near today, but next year in 2021, in October, we will be having this event at the Wageningen University and Research in the Netherlands. So this is just a heads up that you save the date for October 2021 for the next IPPS. Then we have the EPPN 2020, which is a European grant project that organizes and enables people to have access to over 31 facilities, phenotyping facilities all across Europe. Unfortunately, this project, which was very successful and had a lot of feedback or a lot of resonance among the plant phenotyping community, especially here in Europe, of course, is nearing the end. So currently, they are running their 6th call, but of course, as I said, this is the last call for people who are interested to apply. Please visit the homepage the link is indicated down below, and reserve your slot in one of over 31 facilities across Europe to do your experiment. Your experiments will be fully funded, even travel reimbursement will take place, and of course you will be perfectly accommodated at one of these 31 facilities. So when EPPN 2020 is ceasing operations, this is the time when emphasis will follow its footsteps on a much larger scale, concentrating on granting and organizing access to facilities inside of Europe, but also some of which are outside of Europe, and will put this, the work of EPPN 2020 up to a new level. Emphasis will thereby develop, also further develop plant phenotyping infrastructures in Europe. They provide, as I said, access to multi-scale phenotyping facilities, maybe in controlled environments, but also in intensive or extensive field in the space of data and computational service and also modeling. So in China, we are partnering with the AgriX Forum, which was formed as a response to the recent corona outbreak, and serves a similar purpose as our phenomics webinars, to maintain contact to, in this case, Chinese plant researchers. They are currently having a user base of around 5,000 people, of which many are attending the plant phenotyping and phenomics webinars. If you want to go via this platform, then you can use the small QR code that we display here. And the AgriX Forum has been organized by our IPPN member Phenotrade and the 3 other companies which you can see on this slide here. So thanks also and greetings to China. We are very happy that you are here, and I hope you enjoy the show. Last but not least, of course, we want to thank the University of Nebraska-Lincoln, but especially you, Jennifer Clark, who is assisting me here with the moderation, and who is keeping an eye today on the chat, collecting your questions, looking for or after anyone who has problems. So if there is anything maybe during the webinar which needs further explanation, then feel free to post it in the chat section, and Jennifer will pass this information, channel this information back to me, so I can ask that right away. So much, so far so good. So now we are nearing Nadia's presentation, but I want to introduce Dr. Nadia Al-Tamimi a little bit ahead of her talk. She is a recent PhD graduate from Professor Mark Tester's lab at the KAUST, King Abdullah University of Science and Technology in Saudi Arabia, where she studied the genetics of salinity tolerance and rice. During her studies, she had the opportunity to gain experience in high-throughput phenotyping, data collection and interpretation, and also big data management and visualization. She has made collaborations with various institutions, renowned institutions such as Africa Rice in Senegal, or the International Rice Research Institute in the Philippines. Currently, she works as the postdoc at the University College in Dublin, Ireland, and has gained additional experience now going to the field, monitoring experiments there, and of course gaining more and more experience in the space of plant phenotyping, data management, and of course experimental design, big data processing. I'm very happy to welcome her today, and I would now keep my mouth shut and give over the microphone to Nadia.
Thank you, Philip, for that introduction. And thank you everyone for joining. It's a pleasure to be here. I just need a moment to start sharing my screen and my slides. Let's see.
There we go.
Very good from the beginning. And yes, okay, so today I'm going to talk a little bit about part of my PhD project, which was actually the first chapter of my PhD. I obtained my PhD in Saudi Arabia at King Abdullah University of Science and Technology, and the title of my talk is Identification of QTLs Associated with Growth in Transpiration Measurements at the Plant Accelerator in Adelaide, Australia. So other than the fact that rice is a model species with a relatively small genome, rice is a major food crop that feeds over half of the world's population. 90% of rice is produced in Asia, and over 70% of the world's poor is in Asia. So this map shows that each red dot represents 250,000 people living on less than $1.25 per day. So if we want to overcome problems of poverty and hunger, rice has to be part of the solution. And also the global demand for rice will continue to increase due to the expected population growth and decline in rice area. So rice is much more than a grain, like it's shaped cultures, it's fueled economies for thousands of millions of people and been subject to science and traveled all over the world for commerce. So the way rice is produced will need to be altered. Fresh water is becoming scarce due to climate change, and lands are increasingly affected by abiotic stresses, and vast amounts are becoming majorly affected by soil salinity. So this map shows here the saline soils in pink, so in southern Australia, and specifically in the Middle East, so that's why it's been like quite a big topic for Saudi Arabia, and that's why I'm studying soil salinity, and also in places in North Africa, including Senegal. So do rice and salt go together? They might go well together on a plate, but in the field is a completely different issue. So salinity is a major stress that severely affects global rice production. So like we have yield losses of over 50%. And the estimated annual costs yearly is $11 billion US. And rice, from this graph, we can see that rice is one of the most salt-sensitive cereals. So we need to use land that is currently unsuitable for rice production, so that is making salt-tolerant rice varieties in order to meet global population demands. So when plants are exposed to salinity, their growth is immediately reduced and they will produce fewer tillers, and this is the so-called osmotic stress, which is also known as a shoot iron independent stress effect. And this typically includes rapid reduction in turgor, stomatal closure, and reduction in the rates of transpiration, photosynthesis, and growth. And then over time, sodium chloride accumulates in the cells of the shoot, resulting in premature leaf senescence and a further inhibition of growth, and ultimately leaf death, and this is known as the ionic phase. And then in rice, there are two growth stages where salinity is quite sensitive, and that's in the seedling stage and the reproductive stage. So during my PhD, I did study both of these stages, but at the TPA, I mainly focused, so at the Plant Accelerator, mainly focused at the seedling vegetative stage. And then some of the effects that you could see is stunted growth, premature leaf senescence, and in the reproductive stage, you would see grain sterility and yield losses. So in the literature, there are at least 3 separate mechanisms that are involved in determining the responses of plants to salt. The first one is the ion exclusion, so the exclusion of salts from the root uptake. And then there's tissue tolerance, where high levels of sodium chloride are compartmentalized in subcellular compartments such as vacuoles. And then we have the osmotic tolerance. And this is done normally through water conservation responses or upregulation of active uptake pathways. And there have been some studies where they suggest that sensing or signaling molecules might be involved in this type of tolerance. However, if we go back to the definition of salt tolerance, it's a measure of the maintenance of desirable plant traits under salt stress relative to control conditions. And then for application purposes, salt tolerance should be assessed according to the final trait of interest, which is normally yield. And these traits, yield is quite a complex trait, and there are many contributing factors. So then normally we'd study the maintenance of harvest index or maintenance of transpiration use efficiency that all depend on each other for the purpose of the overall final trait. With eye exclusion and the 3 main tissue tolerance mechanisms. So all these types of maintenance mechanisms depend on one another. So in my studies, I use a forward genetics approach. So we have 2 main types of data. So we have the phenotyping, and I phenotype 2 race diversity panels that I'll go on further in the next slides. But the indica and AUSH panel in controlled conditions. Today I'll mainly be talking about controlled, but I did study them in field as well. And I'm looking at certain traits of interest. And the genotypic data is a 700K SNP chip high density array from Susan Mukusha's lab. And we take these 2 data and we do a genome-wide association study. So we examine SNPs across the genome as they represent a promising way to study complex traits in which many genetics variations might contribute to it. So we're trying to identify loci with traits contributing, in this case, to salinity tolerance. So what do you need to build a GWAS experiment, this type of forward genetics? So you need to consider the genetic resources. So I'm using a diversity panel. You can use a NAM population, world relative land races. These are the things that you need to consider. And then what type of phenotyping platform are you using? Are you using the conventional methods or using high throughput methods in the field, or is it in controlled? What type of imaging technologies, depending on what the traits of interests? And are you measuring it through time? So are there multiple measurements, non-destructive? And then for the genotyping technology, what SNP pipeline are you using? Are you imputing SNPs? How are you going to calculate the kinship matrix and population structure? And one of the things that I also focus on in my PhD is a statistical phenotypic analysis. So what's the best way to estimate traits? What type of growth curves should we be using? Is it exponential, linear? Is there another way better to represent it using splines? Should we use the means of the genotypes or the actual individual raw data of the plants? Should there be, and if we have outliers, should we remove them? How important is heritability estimation? And do we need to spatially correct for our data? And then I also focus on what the GWAS analyses. So is there a way to improve mixed linear models? Like, should GWAS be more tailored depending on the type of data you have? So here I'm going to be talking about a proposed model called the interaction model. So what is a GWAS model? So we know that the phenotype equals to the genotype plus its environment. So the basic GWAS model is that you have the phenotype, which could be height or yield, and then we have the marker or SNP, and the environment would be the field, and then you always have this unexplained effect. And this is just the very basic simple of a linear model. And the germplasm I used was funded by the Global Rice Science Partnership, so the GRISP Consortium, which is the PRAE panel, which is Phenomics of Rice Adaptation and Yield Potentials. So there are 2 rice diversity panels, which is that I focus on, the indica and AUSH, which are the total of 559 genotypes that were studied under control and saline conditions at the plant accelerator. So why these 2 panels? Because they represent subspecies level genetic diversity that includes land races of different geographic regions. There are mega varieties that are cultivated in vast areas across the globe, and the diverse and genetically stable breeding material that were nominated by breeders at Erie, CIRAD, and collaborating partners. So the project aim, I mentioned previously, is to identify loci contributing to the responses of rice to soil salinity under controlled condition, just using GWAS. And I do this by studying previously uncharacterized traits, such as transpiration use efficiency using high-throughput phenotyping methods. And then I also had the goal to advance current GWAS methods by exploring improved upon mixed linear models. So yeah, within the first month of starting my PhD, I went to Australia in Adelaide, at a place called the Plant Accelerator, and this is a high throughput facility that can hold up to 2,400 plants, and it's automated non-destructive quantitative measurements, and it's automated data processing. So you can see that these are all of my rice plants. They get put onto these conveyor belts, and they go through this hall where they get weighed in water daily before they enter an imaging hall. And then the next slide will give you more detail. So I used 2 smart houses, which is the northeast and the northwest. So again, you can see here the conveyor belt systems. They're connected to 1 imaging hall. So they go out daily to this imaging hall with 5 camera stations for daily non-destructive phenotyping. So I mainly use the RGB, but there's near-infrared. There's near-infrared imaging for pots and far and thermal imaging. So I'm not sure if it's quite clear, but in this setup, you can see red dots within the plants, the green plants, and these are evaporation cuts. And they were used to determine the spatial variation of evaporation within the smart houses and to calculate the transpiration of plants. So also their locations were chosen to minimize the overall distance from plant cuts. We also added for this experiment here 3 humidifiers in each room. So we can maintain a humidity level that's appropriate for our rice, which I think around 66% humidity. And these were the watering and weighing stations. So next slide. Also, when we did look at the data initially, we could see that there was some spatial variation that's coming from the northeast, where there was some shading because of the hill and actually a tree. So then we thought about the importance of spatial analysis. So each plot, spatial analysis is just showing that each plot does not contain totally independent information, as neighboring plots would affect each other if a plant is taller than the other one, and so on. So we have to consider this spatial arrangement. And we also model this by, it's just a mixed linear model as well, by autoregressions. So we're assuming that plots close together are more closely related than plots further apart. And we can visualize this in something that's called a variogram. So this is just an example of one of my variograms. And what you're trying to do is try to make it smoother without losing any variation. So also I used a split plot design. So which includes a main plot and a subplot. The main plot contains 2 consecutive carts of the same accession, and the subplot is the stress treatment, so salt and control. And also here you can see the evaporation carts that are spaced out within each lane. And then this is just showing the setup. So the system is it's like a pot within a pot system, and we're supposed to simulate the waterlogged conditions of rice fields. So we have a blue pot within a larger white pot, and then we have a specific watering level that we monitor. And then we also added gravel, white gravel, that's to reduce the evaporation of the soil. And then on top of that, blue PVC, and that will help us with the image analysis later. So because you don't want green algae growing on when you're trying to extract green pixels. And then we did the salt treatment at 24 days after transplant with a final concentration of 150 millimolar sodium chloride. And we monitored the plant over 13 days after stress. The next slide is just a time lapse. So I just chose 2 plants randomly with salt and control, and we're monitoring them over 13 days. And you can clearly see that the salt plants at the end of the salt treatment are much smaller. Okay. So we know that the plants are moved towards a stationary camera setup where there are 2 RGB images are captured from a 90-degree angle rotation, which are 2 side views and 1 top view. And then during this process, a pipeline grid is applied to differentiate foreground, which are the green plant pixels, from the background, which are the white pots, the blue frames, the PVC mat that I talked about, and the conveyor belts. And then from this, we can estimate the projected shoot area, which is a sum of all 3 of the green pixels. And then at the end of the experiment, we did some destructive harvesting, and for fresh mass and dry mass, and we can see that there's a strong correlation, positive correlation between the fresh mass and the projected shoot area. And this shows that the projected shoot area, or the pixel count, is a good estimate for plant growth. So some of the key traits that I'm going to talk about, or that I focused on during this experiment, were growth-related traits. So we have the projected shoot area, which is just the sum of all the green pixels, and then the absolute growth rate is the total growth per unit time. And then we have the relative growth rate, which is a prominent indicator of plant productivity, and it's just the increase in size relative to the size of a plant present at the start of a given time interval. So we're just, in this case, the day before. And then I looked at water loss data from the weighing and imaging. So from that we managed to estimate transpiration, which is the difference between the weight before and the weight after, subtracted by the evaporation cuts. And then from that we can calculate the transpiration rate, so that's just over time. And from these 2 growth and water loss data, we estimated transpiration use efficiency, which is the ratio of biomass produced per unit of water transpired. Okay. And then just how we estimated some of these traits is that we looked at the projected shoot area for, here we're showing an example of the indica panel, the grand average, over the 13 time intervals. And we can see that the control does follow an exponential growth, but that's not the case for the salt, so it's neither exponential nor logistic. So how could we find a better way to visualize this data? With the help of a statistician, Chris Bryant, we were able to accurately describe plant growth and to better estimate a PSA. So we fitted cubic smoothing splines, which is a piecewise defined polynomial function. And this approach has the advantage of making no a priori assumption about the shape of the curve, and it gives an unbiased analysis of high-throughput phenotypic data. And here, each line is showing each individual plant, the spline for each individual plant. And then we saw that looking at the relative growth rate, the spline data of the relative growth rate, rice shows a salt response in 2 main intervals. So the first one is in interval 2 to 9 days after salting. So we can see a rapid decrease in the relative growth rate between these 2 time intervals. And then the decrease kind of plateaus after 9 to 13, in the interval 9 to 13. And then 2 to 9, we further split as RGR declines less rapidly under saline conditions when compared to the 6 to 9. And we also did this to kind of relieve computational power for the GWAS analysis. So instead of doing a longitudinal daily analysis, we decided to also split them up into intervals. And then looking at the trait of interest, which is the transpiration use efficiency, which again is defined as the ratio of above ground biomass produced per unit of water transpired. And then you have to realize that this is quite a complex trait because it's a third order derived trait. So it's estimated from transpiration and growth, and this is in turn estimated from the pixel count and the water loss. So here, when comparing the indica and Auschwald, the average, on average, TUE decreases decreases more rapidly for salt-treated plants, and the indica panel had a lower average decrease in TUE when compared to the Ausch. It was 16% compared to a 24% decrease. And then to quantify the relative performance of plants with respect to TUE, we used a simple index, which is just the ratio of salt-treated plants relative to TUE in control plants for that same time period. And we represented it in a box plot here. And this shows that the indica tends to maintain higher TUE than the Ausch panel for the first 3 intervals. And then the next part is the GWAS. So we compared 2 GWAS models. The first one I like to call like the conventional mixed linear model, and we just used the kinship matrix. And this was done in Tassel. You can also do this in Gapit in R. And then we used the proposed model is known as the interaction model. So we have the treatment, which corresponds to either salt or control, and the marker, and then this is the extra covariate, which represents an interaction term between the marker and the treatment. So it's like the SNP effect in response to the treatment, control, or salt. And this is done with an ASRML package in R. And then to determine whether different loci were associated with trait variation in different diversity panels, we use the indica and Ausch independently, and then we use the combined, which we're calling the IndAusch. Okay, and this is just to kind of show you the difference between the marker and marker treatment interactions. So the marker term suggests that any loci contributes to the traits regardless of the treatment, whereas significant loci that are responsive in this part are responsive to salinity treatment. For those of you who might not know this is the output of the GWAS analysis, which is known as a Manhattan plot. So you have the LOD scoring and the chromosomes. There are 12 rice chromosomes. Each dot here represents a SNP marker. And then we have, this is the Bonferroni threshold, and anything in green or above this threshold is a significant SNP. And then further looking into the GWAS analysis, we saw that rice shows a dynamic response for transpiration use efficiency. So we identified some loci present exclusively only during the early time intervals on chromosome 11 after salt treatment, and then the presence of that loci would disappear in the next interval, and then you could see something significant possibly popping up in chromosome 3 and then disappearing altogether in the later phases. So thus rates exhibits a dynamic and complex response to salt stress that's consistent with the hypothesis that there are two distinct phases of salt stress. And then when looking at in depth of the SNPs and trying to narrow down candidate genes, our results showed that the candidate genes found in the early intervals are mainly encoding for signaling proteins, further supporting the hypothesis that the early response to salt stress, which is the osmotic phase, could be related to signaling mechanisms. And then in summary, we used cubic smoothing splines, and it was the best way to estimate plant growth and transpiration. We identified 4 time intervals that characterize the early response of rice to salinity. The main traits that we analyzed, RGR, which is relative growth rate, transpiration rate, and transpiration use efficiency, was analyzed using a new interaction model. We could see that rice shows a dynamic response to salinity, and the candidate genes suggest that the early response to salt stress could be related to signaling molecules. So like in conclusion, we uncovered new sources of salinity tolerance by identifying loci associated with growth, transpiration, and yield. Well, and that could be for another time. And we estimated previously uncharacterized traits, like especially in respect to transpiration. We improved current association models. Here I talk about the interaction model, but further later on in my PhD, I propose a one-step GOS model that can also be for another talk, but it's essentially that I'm using the raw data, so each individual plant, instead of taking the mean or the median for GWAS, and we're putting it and doing the spatial correction all in one step. And the whole point of my PhD or my work or my projects are to develop a holistic approach that recognizes the complexity of genotype and phenotype interaction. So some of the limitations in the future perspective is that I use the 700K SNP data. There is now a 4.8 million SNP data, so it'd be interesting to run GWAS on that and see what kind of significant SNPs or hits I get for that. There is, from this, the TPA, there is a wealth of data. I'm only showing you 3 traits. There is like plant compactness, like loads of architectural traits that I need to dive into, and validating the candidate genes underlying this interesting loci. That in itself is just a whole nother PhD. And this is just a short sketches at how my PhD is, that I took years to collect data and years to just interpret what's the best way to statistically estimate, because again, we're using proxy data, imaging as just a proxy for plant growth, things like that. What's the best way to represent it, and then writing it up. And I feel like at the end, all I got was like one Manhattan plot or a couple of Manhattan plots. And all of this work is published in Nature Communications, and all the data from which the traits were calculated, the trait values, and all the code used to estimate the traits, and the code for the interaction model are all available online. So if you're interested, please go check that out. And now what I'm doing now, so my high throughput phenotyping journey is that in Saudi Arabia, I mainly focused on the controlled conditions, although I did have field trials, but they weren't high throughput. And then I moved to, in January, to Ireland at UCD, where I'm doing high throughput phenotyping in the field. But I'm not studying salinity tolerance for the seasons of Ireland. Due to the seasons of Ireland, I am actually looking at another abiotic stress, which is waterlogging in Bali. So, and this project is funded by the SFI grant, Science Foundation Ireland, and the title is Looking into Time and How Abiotic Stresses Impact Barley Production and Malting Quality. So the research aim is to assess genetic diversity, perform detailed physiological characterizations under control and stress conditions, and study the effects of waterlogging in molting quality. So currently we've got 2 ongoing field trials at the moment, and we're using the panel called the Exhibit, which is European Heritage Barley Collection. So there are about 400 lines in the field at the moment. And we're starting to characterize those 400 different varieties of barley. So heritage barley is underutilized and understudied genetic resource. So our hypothesis is that modern cultivars were bred under optimal conditions, favoring yield traits, while the heritage germplasm, I think including 9 Irish heritage lines, have higher stress tolerance but remain largely uncharacterized. So I'm going every other week to the field and taking UAV images, and also we'll deal with hyperspectral data, which should be fun and interesting, and looking forward to that. So it's quite similar to what I did for my PhD, but it's just to remind you that it's quite an interdisciplinary research. Because there's high-throughput phenotyping, there's genomics and genetics, and there's also the computational aspect. There's also statisticians, so it's definitely a collaborative type of research. And I'd just like to thank my my PhD advisor, Mark Testa, Sonia Negrao, Helena Oki, who has been a big help, who taught me all of the spatial statistics, and just been a big help throughout my PhD, and all the people at the Plant Accelerator, Bettina, Chris, Brian, Lydia, Susan McCouche for providing the SNP data, and all the germplasm were provided by ERI and the Gene Bank, and my supervisor at UCD now, Sonia, and the SFI. And thank you, and I look forward to taking any questions.
Thank you very much, Nadia. What a tremendous piece of work. I mean, that although it's very common among phenotyping experiments or phenotyping research, if you put it into application, you just have to touch a lot, as you said, a lot of different disciplines, going from computational, quantitative genomics to experimental planning. And especially if you're now doing this in the field, this certainly will add a new layer of complexity to this already very complex work. So heads down for you. Fantastic talk, fantastic work. I mean, it has been published in Nature Communications, and it's a terrific piece of paper. So if you find the time, I would really love if you could post a link into this, the chat section, or if you could provide me with this link so I can upload it to your site, so that people can find and cherish this work.
Yeah, definitely.
Thank you very much. So we got a couple of questions from the audience that were flying in when you gave your talk. I will just start with the first one and then go by the sequence that they arrived here at my desk. So first question was from Mr. Singh. He wants to know what was the vapor pressure deficit of the greenhouse for where you cultivated the rice? Do you know?
Well, no, I'm actually not sure of that question. I might have to go back to the paper if it with all the details. This experiment was done in 2015, so it was really the first project of my PhD.
Is it in the paper somewhere in the because Jennifer just pasted the link into the comment section. So Singh, if you want to dive into it, you might find the answer to this question in that paper.
Yeah, let's check.
Okay, so next question came from Jennifer Clark. She asks, so ASR EML is a mixed or fixed and random effects model. So
Yeah.
what terms are fixed and what terms are random? Can you elaborate on that? Very detailed questions. I have to say, Jennifer is a professor for statistics. So.
Oh, okay.
Sorry.
No, that's fine. So the fixed effects was definitely treatment, because that's normally fixed, and the random effects was mainly when we also did the spatial analysis, was the rows and the columns. So I don't know if that
they
kind of
accounted for the spatial heterogeneity there.
Okay, yeah, I was just curious whether if the genetics played a role in the model, because you're doing a GWAS, so you're saying the GWAS response is fixed.
Mm-hmm.
Okay, that was all my question. Thank you.
So next question comes from Prakriti Pudyal. Sorry if I mispronounce the name, but I can only read it here. He or she asks thanks for the nice presentation. Did you also measure root characters under salinity?
No.
And if, what?
Yeah. I wish I did. I also wish I took sodium and chloride at concentration, but no, I didn't look at roots at all for this project.
All right. Thanks. Then we have a couple of questions all going in the same direction. How much heritability could you observe for those traits that you observed so far?
Yeah, so actually this is something that was not in the paper, the heritability, that I did calculate it properly for the field experiments, but looking back when I did calculated the heritability after the publication, that they were enough. They're about 40 to 60% heritability, which is sufficient enough for a GWAS, I think.
Okay, and that was based on the relative growth rate and
yeah, for all three traits, and you could see that the heritability also improves after you do a spatial correction. So I checked the heritability before and after, and that's why also it's important to maybe consider spatial analysis, because it could increase your heritability. Okay.
A small note going out to our Chinese audience. We do not see you here in our chat. So if you have questions, please speak them out or write them in the AgriX forum, and members here or members of our auditorium here will then channel them over to me. Yeah, so don't hesitate to ask. Although both platforms are not directly linked, we have a few people here and there sitting that can channel the questions from China over to here. Please ask questions in case you want to ask them. So going back to the questions from our auditorium, there's one from Lee asking, are there any different results between two models, main effect model, interaction model? Did you observe them individually?
Yeah, so I did look at them individually, and I didn't include the main effect or even the conventional model because there was no significant loci, at least when we used the stringent type of corrections for the Bonferroni, the FDR. So it was just, and with the interaction model, we managed to get significant peaks.
Mm-hmm. Thank you very much. There's one question from Paulo Herman. He asked, did you try to measure transpiration or transpiration rate in the field conditions or just in your smart house?
So for this one, just in the smart house. So in the field, we were mainly focusing on yield related traits, but I did kind of inherit some data from IRI, the International Rice Research Institute, from 2013, and they had some transpiration data. that I worked on. But when I tried to correlate it with this control data, I couldn't find like strong correlations for transpiration. But no, it wasn't like one of my experiments that I tried to translate it to the field. In the field, I mainly looked at a yield.
So this question coming from me, after you did your transpiration experiment or your water use experiment, what was the interval or the timely frequency in which each single pot was measured? So I mean, you showed this tremendously big high throughput phenotyping system that you used for your studies, and yeah, it looked like around 1,000 individual pots per side of the greenhouse, northeast, northwest, I think you said. And what was the throughput in terms of measurement? When at what frequency was a single pot recorded for transpiration rate or for water loss?
So it was done. So the whole, for it to go around the conveyor belt each day and get weighed and imaged daily for the whole 2 greenhouses took 8 hours. And so we also have the timestamp data for each session, and so that's also correct. So we also corrected for transpiration throughout the day, because obviously it's going to change with the daylight. So we also took that data into account. But yeah, for the whole process, I think it took to image and weigh those plants every day. It was within an 8-hour time frame.
All right. Would you say, from your personal viewpoint, that you would you maybe be able to derive further insights into the regulation and dynamics of transpiration if you would have had a constant real-time tracking of transpiration? And is there any ambition to investigate the transpiration reaction at a smaller time scale?
Yeah, yes, definitely. I think now there is a postdoctoral student, postdoc who's taking over this work, and I think they're going to do another experiment just looking at low and high transpiration rates of individual plants, and they're setting up a different experiment. I don't know if I'll be involved in it or not, but it's definitely something that our lab is looking into.
Very good. Thank you very much. Then, yeah, of course, there are several people that want to contact you directly to speak and continue to speak with you about your work. In case you want to make this here more or less publicly, you can write your contact details, of course, into the chat. Otherwise, I would say you certainly have at the university college where you are working now, you have a work email address. Are you fine with people sending you further questions via email?
Yes, of course. I do welcome any questions.
They can find it at the UC Dublin.
Yes, I definitely am on their website and also on the CAUST. I can also share my email address here to all the panelists.
Yeah, that would be great. Yeah, there it is. So, Jennifer, how are we on in terms of timing?
We're fine. We're at close to an hour. We've addressed all the questions. The only remaining question I see is the attendees would like to know, oh wait, there's another one. This was a question we missed. Are there any connections between your past and current work? Like, salinity and waterlogging stress, are those related, especially in connection to root growth and above ground plant development?
Okay, yeah, sure. There is some similarities because they're both abiotic stresses. I can't say much about root because I haven't really studied or don't have that expertise for root development. But they have similar, like you can see reduction in growth, you can see early senescence. But one of the differences, at least that I'm encountering in this field trial, is that normally in salt stress, when it comes to flowering, they flower early, but in waterlogging, I'm seeing the opposite, they're flowering a bit later. So there are some similarities and differences, but the work is kind of the same. So I'm doing phenotyping, doing genetics and GWAS.
Great, thank you. Philip, the only thing left was a request to let the attendees know where the recordings are posted of the previous webinars, so where that information can be found.
Yes, I just posted a link to our IPPN YouTube channel. You will also have the opportunity to go via the IPPN homepage and the Phenomics there, the Phenomics webinar subpage. There's always, for the ones that are already uploaded, the direct link to the YouTube channel, or just use the link in the chat. For any further questions, please don't hesitate to go on our IPPN homepage. It's www.plan-phenotyping.org. There you can also find contact information. So in case there are further questions, just drop me a mail and I will respond to them in a timely manner.
Perfect. Okay, so one more question for Nadia, which is, has there been any validation of the QTLs or genes that you identified in the study? I guess follow-up is, has the work been linked to any development of salt tolerance lines in rice?
So yeah, there is follow-up work. It's still at the beginning stages of this product. So the lab at CALS is now preparing RNA sequencing data using accessions that are showing like high or low maintenance for TUE. They're also working on transforming for promising candidate genes for functional studies. But again, it's still in the early phases, and yeah, hopefully once something develops more, we can share that with you.
We'll be definitely keen to hear more from you, more from your ongoing work. And yeah, I would also, from the side of the organizers, like to thank you, Nadia, for this very, very interesting work. And all the best for the future then. And your
Thank
further
you.
Great, thank you, Nadia.
Thank you.
Yeah, we are almost at the end. I just wanted to hint at our next webinar speaker, which will be Dr. Jörg-Peter Schnitzler from the Helmholtz Center in Munich, in Germany, and he will be telling us a little bit about the secret smell of plants. So how can we use this for plant phenotyping? It will be all around rather, yeah, underrepresentative parameter and in volatile organic compounds of plants, and hopefully shed light into how plants communicate with the environment and with other kin. So I'm very, very excited for this talk, as well as I was excited for this today's talk, and I will be happy to see all of you and more next time in 2 weeks at the same time slot. Thank you very much.
Thank you, Philip. Thank you all attendees. Thanks, Nadia.
Thank you.
Bye-bye.
Bye-bye.